audiencia, las personas que nos acompañan esta mañana. Muchas gracias por estar con nosotros. La Corporación Académica Ciencias Básicas Biomédicas les da una bienvenida a este evento académico organizado en el marco de la conmemoración de los 25 años de la corporación. En esta actividad contaremos con la presencia del comunicador de la ciencia, Richard Harris, quien nos presentará su libro Rigor Mortis, de cómo la ciencia mal hecha crea falsas curas, mata la esperanza y malgasta billones, el cual ha sido traducido al castellano por iniciativa de Foros COVID-19 con la financiación de la Vicerrectoría de Investigación de la Universidad de Antioquia y editado por el Fondo Editorial Biogénesis. El traductor de este libro es el profesor Gustavo Zapata de la Escuela de Idiomas del Posgrado de Traducción de la Universidad de Antioquia y el editor de la versión castellana, el doctor Jorge Osa Londoño, quien nos acompaña para hacer una introducción de nuestro invitado y guiar esta presentación. Profesor Jorge Osa Londoño es médico veterinario con maestría en virología en la Universidad de Wisconsin y doctorado en microbiología de la Universidad de Virginia. Muchas gracias, profesor, por acompañarnos, por introducir este evento. A todos ustedes, muchas gracias por estar con nosotros y por acompañarnos en el marco de la conmemoración de los 25 años de la Corporación Académica Ciencias Básicas Biomédicas. Les damos la bienvenida, profesor Richard, muchas gracias. Profesor Jorge, lo dejo a usted en la presentación inicial.
Bueno, muchísimas gracias. Muchas gracias a todos. En primer lugar, pues a toda la audiencia académica de la Universidad de Antioquia y también del resto de Colombia y en el exterior que han aceptado esta invitación en celebración de los 25 años de la Corporación de Ciencias Básicas Biomédicas. Muy especialmente este saludo va para los estudiantes, para los egresados y para los profesores de la corporación. Justamente ellos son pues el presente y el futuro de esta institución. Voy a repetir un poco para ser un poco más amplio con los créditos de todas las personas que han colaborado para esta iniciativa. Since the speaker will do his presentation in his native language, let me switch into English now. This academic event for the celebration of the 25th anniversary of our corporation is the product of Foros COVID-19, which is an initiative of the School of Microbiology and Bioanalysis in cooperation with the School of Public Health, both at the University of Antioquia and under the leadership of Professor Alonso Jaramillo and the international advice from Dr. Juan Carlos Zapata. Excuse me. Excuse me, in one of those forums, we discussed the subject of infodemia and its impact on the pandemic. And for that moment, we had the happy opportunity to have the participation of Richard Harris, whom we had met from his book, Rigor Mortis. It was here in this forum where the idea of translating the book took shape. Alonso from the School of Microbiology would try to get the budget from the Vicerrectoría of Research. Professor Gustavo Zapata from the School of Languages would do the translation, and Jorge Osa would do the editing of the Spanish version, and Dr. Lina Carrillo, the director of the editorial group Biogenesis from the School of Agriculture, would do the final job. Everything was successful, and the job was accomplished in a record time of 5 months. Let me now present Richard Harris and his book. He is a recent retiree from the U.S. National Public Radio, where he served in the area of science, medicine, and environment for the last 30 years. He has been the winner of three times of the national awards from the American Association for the Advancement of Science. And his book, which has also been translated into Japanese, was originally published in 2017, and it deals with a profound and very well-documented reflection on the major errors of basic biomedical research, particularly at the academic institutions. He also dwells on the causes and the impact on society, as well on the necessary steps that it is necessary to undertake to find the road again. Among other things, changing the curricula for the education of new scientists, particularly going back to philosophy and interdisciplinarity, and definitely opening much more space for thinking. And permítanme make a digression here, porque yo considero que este párrafo es muy importante y quiero que pronunciarlo en español para que ustedes mantengan el interés a lo largo de la charla. Este último párrafo, entonces, se los quiero decir en español. El libro, que también ha sido traducido al japonés, fue publicado originalmente en 2017 y se trata de una profunda y muy documentada reflexión sobre los errores mayúsculos de la ciencia básica biomédica actual, particularmente la que se realiza en las instituciones académicas, sus causas y sus consecuencias sobre la sociedad. Además, presenta indicaciones y recomendaciones acerca de cómo volver a encontrar el camino, entre otras cosas, cambiando la estrategia en la educación de los nuevos científicos, retomando la reflexión filosófica y la interdisciplinariedad y definitivamente dedicando más tiempo, mucho más tiempo al pensamiento. The final objective of the book is to try to keep science as the most efficient method to conquest the deepest secrets of nature so that it maintains the utopia of helping to construct a better world. Now then, I pass the word to Richard. At the end of his talk, I would ask some questions and open up the microphone for all others who may ask questions to Richard. Thank you very much.
Thank you. Let me see if I can get my screen shared here. Can you see my screen?
Yes.
Yes.
Great. Thank you. I'm pleased to be with you today. Thank you for inviting me, and thank you for helping spread the word about rigor and reproducibility in biomedical science by publishing my book in Spanish. While I will spend my time today talking about problems with research, I always remember how important biomedical research is. We have certainly seen it working at its best during the coronavirus pandemic. My book focuses on preclinical research in biomedicine, and I won't just talk about problems, I will also talk about solutions. To give you an example of why this is important, I would like to start by talking about a man I met a few years ago. Tom Murphy had been diagnosed with ALS, a neurodegenerative disease that is always fatal and lacks good treatment. Mr. Murphy wanted to get into a clinical trial for a potential new drug. He looked around very carefully at his options, and he finally chose to sign up for a clinical trial of a drug nicknamed Dex. He felt that the drug helped him, at least for a while, but overall, the drug provided no overall benefit to the people in the trial. And as it turns out, Dex is not the only drug that has failed for ALS. There is no effective treatment. A research group in Cambridge, Massachusetts called the ALS Therapy Development Institute decided to look into that dismal track record to understand why. They rounded up the preclinical studies in mice that had led to the development of these drugs. These mouse trials are typically used to help identify drugs that might be useful in human beings. As you can see on this slide, it seemed like quite a few of these drugs could work, at least in mouse studies. By the way, DEX is the bar at the bottom, showing an increase in survival time by a bit more than 10%. But most of these animal studies were based on tiny samples of mice, sometimes just around a dozen animals. Sometimes the researchers didn't bother to separate litter mates, so you might have litter mates together in the control group and a different litter in the test group. This is a serious design flaw because differences between the groups could simply be due to biological differences in the mice. The ALS Therapy Development Institute decided to run really careful new studies of all these drugs to see if the studies were done right. Here's what they found. The results from experiments conducted by the ALS Therapy Development Institute are the black bars. You can see that when these studies were done properly with enough mice and carefully controlled, essentially none of them worked. Unfortunately, most university scientists simply can't afford this kind of experiment because each test costs over $100,000 when they are done right. So very often a university researcher will run a study with a very small number of mice. They'll call it a pilot study and hope that the pilot studies actually reflect reality. And as you can see, that's not true. This general problem came to the attention of a scientist named Shai Silberberg at the National Institutes of Health here in the United States. His job was to help hand out federal research funding for clinical trials of drugs to treat ALS and other neurological conditions. He noticed that the committees that allocated these dollars never even stopped to ask about the quality of this preclinical research. They assumed it was solid, so they were focusing entirely on the design of the clinical trial itself. He started asking questions about this. As a result of his questions, the NIH started asking hard questions about whether the studies they fund are reproducible. Other scientists started asking questions too. The lead author of this paper, which was published in 2012, is Glenn Begley. He was the head of cancer research at the drug company Amgen. He decided to take a second look at 53 published papers he thought could have been the basis for important drugs if they had actually worked out. They were exciting ideas, mostly from university labs, that Amgen picked up but dropped because they couldn't replicate the findings. Begley enlisted the aid of many of the original scientists to attempt replication studies of these papers. In the end, they could only replicate 6 out of the 53. That's about 11%, a pretty bad success rate. Of course, this paper itself isn't reproducible because Begley promised the authors of those original papers that they would be anonymous. So we don't know who they are or what their experiments were. So nobody can actually repeat this experiment, but still it raised further alarm about the issues of reproducibility in biomedical research. And a similar paper from the Bayer Drug Company came to the same basic conclusion. More warning signs also came from the top of biomedical research. Here's NIH Director Dr. Francis Collins. To his credit, he decided to confront the issue rather than try to pretend it didn't exist. Here's what he wrote in a commentary a few years back with his chief deputy, Larry Tabak. As we all know, science is self-correcting in the long run, but they wrote that, quote, in the shorter term, the checks and balances that once ensured scientific fidelity have been hobbled. The so-called reproducibility crisis is now recognized throughout the scientists. Here's a not very scientific poll that Nature conducted back in 2016. It finds that more than half of the scientists who shared their opinion said there is a reproducibility quote unquote crisis. Another 38% said there's a slight crisis, and the remaining 10% said they didn't know. I don't actually like the term crisis. That's not really what's happening here. I'd say this is instead the recognition of problems that have existed for decades. I think it's actually good to recognize problems because that's the first step in solving them. Here are some of the key issues and causes of these problems. Later in the talk, I will talk about some of the solutions. One problem is bad ingredients, such as antibodies and cell lines. More about that in a minute. Experiments may also not be planned carefully enough. A good experiment should deliberately seek to reduce bias in the experimental and in the results and in the final analysis. As the famous physicist Richard Feynman said, the first principle of science is that you must not fool yourself, and you are the easiest person to fool. Scientists need to spend more time thinking about their experimental designs to make sure that they are reducing the risk of bias. Another statistical errors also turn out to be a major problem, usually because many scientists don't know any better, but sometimes through deliberate manipulation. One of the most serious problems is the use of p-values in scientific analysis. They have become the bright line of what's publishable and what is not publishable. Not only is that wrong, it also creates an incentive for people to consciously or unconsciously manipulate their findings to meet this arbitrary threshold. That manipulation is called p-hacking. And last but not least, I will talk about the incentives, particularly in biomedical research. Scientists aren't rewarded for doing careful, reproducible science. They are needing exciting results to keep going in what has become a hyper-competitive environment. So when it comes to bad ingredients, there's no better example than Henrietta Lacks. Scientists at the Johns Hopkins Hospital in Baltimore harvested her aggressive cervical cancer cells back in 1951, shortly before she died. Those cells became the world's first immortal cell line called HeLa cells. It turns out her cells grew really well in the lab. In fact, they grew better than just about anything else. So before long, many other cell lines scientists were working on, or thought they were working on, had actually been overrun by these HeLa cells. Biologists have recognized this as a problem for a long time. I read a research paper about this back in the early 1980s. But scientists generally ignored that warning. Nearly 40 years later, it's still an issue. This paper was published in 2017, and it attempted to quantify just how big a problem the misidentified HeLa cells have become. So you can see they found nearly 33,000 studies in the literature that had been done with cells that they thought were from one other cell line, but were actually HeLa cells. And those papers have been cited by something like 500,000 other papers. Practically nothing in this literature has been corrected or retracted. This paper grimly concludes that the problem with HeLa cells is only getting worse. It's essentially contaminating the medical literature. And HeLa is the single worst offender, but it is by no means the only cell line that's running out of control. This group, called the International Cell Line Authentication Committee, has identified more than 400 cell lines that are not, in fact, the cells that they are purported to be. Some of the contaminants aren't even human cell lines. Now, this paper talks about a serious problem in data analysis. The problem is called harking, which means hypothesizing after the results are known. This problem got its name in 1998, as you can see in this paper, here from a psychology professor. Many people don't realize why this is a problem. It seems okay at first to get your data, then look it over carefully, and finally decide what hypothesis your data supports. My favorite illustration of why this is not a good idea is this cartoon. Think of the little girl's arrow as her hypothesis. You can see how easy it is to hit your target if you wait until just after you have your results. Now, this is amusing as a comic strip, but this actually happens all the time in biomedical research. Looking over data is a perfectly legitimate way to generate a hypothesis, but you can't then use that data you've collected to support the hypothesis. You need to design a study from scratch and collect new data. One reason scientists make bad decisions like this is they are in a fierce competition for funding. This graph of funding from the US National Institutes of Health shows that research funding doubled between 1998 and 2003. That led to a flood of new labs and new scientists, but the funding since then stopped growing. So scientists have been struggling to get enough money just to keep their labs going. That creates an incentive for scientists to make their results seem as exciting as possible so they can win the fight for funding. This problem, called publish or perish, is not new. And of course, it's not just in the United States, but the funding crunch here makes the problem much worse. And it's also obviously a problem funding around the world. To measure the phenomenon of this exaggeration that scientists feel compelled to use in their studies, these Dutch researchers took a look at the change in rhetoric in scientific papers over the years as funding pressures increased. In this paper, which they published in 2015, they found a big increase in positive words in abstracts. They found words like robust, novel, innovative, and unprecedented increased by up to 15,000%, as they said. Of course, they were having a little fun in how they presented this data, but the point is scientists really feel they must sell their findings. Their careers depend upon it. It matters more to have this kind of flashy result than to have something that is solid and reproducible. So let's take a minute. What was it? What happened here? So let's take a minute to consider the consequences of this hyper-competitiveness. One thing we see in this graph is that drug discovery is actually getting slower and more expensive. Reproducibility is probably playing a role here. If more results of universities, sorry, I got it. Reproducibility is probably playing a role here. If more results out of university labs were correct, drug companies would spend less time working on ideas that turn out to be bad. Scientific studies will never get the right answers every time. We shouldn't expect that. Scientists are exploring the unknown, but scientists can do more to avoid obvious errors. Scientists have recognized these issues, and some are actively working to find ways to improve the validity of research. One thing they can do is validate their ingredients, such as their cell lines. Another solution involves increasing the transparency of scientific findings, so exciting results can more quickly be evaluated and replicated. Scientists are also increasingly aware of the value of verifying results, and they're also looking for ways to change some of the bad incentives that are at the root of so much of this problem. One step is to be more careful about the antibodies that are used a lot throughout biomedicine. It turns out that many of them simply don't work as well as scientists expect they do. But they can avoid being fooled by antibodies that give wrong answers if they run more controls in their experiments and don't take the antibodies for granted. Transparency is another powerful tool I mentioned to improve reproducibility. One of my favorite stories about this is the value of sharing data as it relates to the original sequencing of the human genome. You may recall this big moment back in 2001 when they published the sort of first more or less complete draft of the human genome. One of the most surprising results reported in this paper was the claim that bacterial DNA had apparently jumped directly into the human genome in recent times. Now, viruses can insert their DNA into ours, but bacteria aren't supposed to be able to. That's why this finding was so surprising. But the conclusion turned out to be wrong, and we learned that very quickly because the genome sequence is put up in a public database for other scientists to reanalyze. This group pointed to an analytical error that had led the original team astray. So here's a perfect example of the value of transparency. It took only months to correct the scientific record on this. Unfortunately, there are far more stories where scientists refuse to share their data so others can check their work. Another step scientists should take is to make sure that the cell lines they are using are correct. This is from a cell line established in 1976. It's called MDA-MB435 and has been used ever since to study metastatic breast cancer. But there is strong evidence that this cell line was contaminated just a few years after it was isolated. And it turns out the cell line is probably a melanoma cell line. Back in the 1970s, it was hard to catch problems like this, but now genetic tests make it easy to verify the cell type. Companies will provide this service for a relatively small fee. The NIH now expects scientists who are using cell lines to run these tests to make sure the cells are what they are supposed to be. Unfortunately, there is no easy way to enforce this requirement, and scientists continue to use this melanoma cell to study breast cancer. Now, I want to end this talk by mentioning some of the people who are trying to change the culture of science in order to reduce the bad incentives that are driving these problems. This professor, Brian Nosek, tells an interesting story about how he got tenure at the University of Virginia. He's a psychology professor, by the way. His department chairman asked him for a stack of all of his published papers. He said, I published something like 200 papers. The tenure committee isn't going to read all of those papers. What are you going to do? Weigh them? And the chairman still demanded that he submit all his papers, so he did. Now, Nosek realized that if he'd come to the University of Virginia and at the very outset he'd been told, when you are up for tenure, we are going to ask you to submit your 5 best papers, that would have changed his incentives as a researcher a lot. He would probably publish a lot less, but he would publish things that are really high quality and potentially of much greater interest. And so he started thinking about different ways that you could change the culture of academic research to try to get the incentives right. And he's making a career around improving reproducibility, at least in part by improving the culture of science. And although he's a psychology professor and he spent some time looking at the problems in psychology, he's also focused a lot, and his group has, on biomedical research as well. And I could talk about more about that if you're interested. Now, Nosek is not alone. David Allison at the University of Indiana is also putting energy into this. Here's a paper he wrote a few years back highlighting the problems introduced by scientific publishing. He tells the story of finding an error in a published paper. He submitted a letter to the editor pointing that out, but he was told this journal would only publish the letter if Allison paid several $1,000s in page fees. Dr. Allison has been raising issues like this to improve science. Another way to change the culture is to change medical and biomedical education. Dr. Arturo Casadevall at Johns Hopkins University is among the people who've been thinking about that. It turns out that biomedical researchers don't learn enough about good methodology when they're graduate students. People, by and large, learn from their mentors, and mentors are more interested in having their students work at the bench rather than taking more courses to learn good methodology. Arturo Casadevall decided that he wanted to try to fix that at his university. So he has been developing curriculum that really tries to get people to think more carefully about experimental design. He says he wants to put philosophy back into the PhD, so scientists he graduates aren't just good technicians. Across the Hopkins Medical School campus from Arturo Casadevall is Barbara Slusher. She works on drug development. She realized that even at Johns Hopkins, one of the top universities in the country, a lot of exciting ideas don't pan out. As a result, drug companies are increasingly reluctant to work with university researchers. So Professor Slusher set up a center that actually seeks to replicate ideas before they are offered up to drug companies. And often she found that the results aren't replicatable, and she sends people back to their lab to try again. And the United States National Academies of Sciences, Engineering, and Medicine has also been thinking about these culture of science issues. They came out with a report in 2017 called Fostering Integrity in Research. It was a long-awaited follow-up to the Academy's work on scientific misconduct. But instead of focusing on scientists who are breaking the rules, this report looks at the system that actually encourages that unwanted behavior. It turns out that people set their standards based on what people around them are doing, and they might think it's okay to take serious shortcuts if that's what their colleagues and competitors are all doing. Of course, solutions require changes at all levels, from the funding agencies to the college deans, department heads, lab heads, and the scientists themselves. And that is not an easy task. Now, it is so important to get the fundamentals right. Medicine is built on a foundation of this preclinical research, so these results need to be as robust as possible. I would argue that most scientists are honest and they want to do work that helps other people. That goal should not conflict with doing what it takes to keep their careers alive, but unfortunately, sometimes it does. Thank you.
Okay,
Thank you.
Richard. This is wonderful to listen to you, besides your very easy to understand pronunciation of English. That's great. Congratulations. But other than that, the ideas, the more I listen, and I think I have read your book several times for doing the work, and I have listened to you also several times, but the more I listen, the more overwhelmed I am with all this information. I think that as we were trying to do, you have to come to Colombia. Let's hope that this pandemic goes quick because there are so many other things that we want to talk to you. But at this time, I have some questions. The first is, do you think that there has been an impact already on academic communities, on graduate schools, on journal editors, on the decision-making groups at all the levels in the institutions and the government?
I think there has, and it's not just my book, of course, because my book is describing a movement that's growing in the United States and elsewhere around the world to address these issues. But the NIH, as I mentioned, has instituted some new rules around things like cell lines, university tenure considerations, and at least in some of the major institutions in the United States are thinking about, wondering if there's a better way to grant promotions and tenure that does not create incentives for people to exaggerate the work they're doing. Journal editors certainly are aware of this issue, and there are some. One of the problems in journals has been that I'm sorry, Jorge, were you going to translate? I forgot I'm going on here. Do you want me to stop to translate, or are we going to do this in English?
No, Richard, I think that your pronunciation is perfect and we all are understanding you very well.
Okay, good. But journals, one of the issues with journals is journals only want to publish very exciting papers, and they often even won't publish negative results that basically that either contradict what's been in the literature before or that find a result that doesn't work out. So, but there are now some journals that recognize that distorts the scientific literature, because if you only publish the studies that seem to work and don't publish the ones that raise questions about that, you create a bad impression about what the science actually means. And so there are now journals such as the PLOS journals that will actually publish results of anything that's good science, regardless of what the results are. And so I think that's a very important step forward as well.
Okay, there is a question here. Richard, thanks for your talk. I would appreciate some words about the use and abuse of animals in science.
This is a difficult issue because animals are expensive. They're also there's understandable reasons not to want to use too many of them, because not only for expense, but for ethical reasons. And so scientists and institutions generally require scientists to use as few animals as possible. And that's humane, and it's very understandable. But at the same time, if scientists end up using far too animals in their studies, they don't get meaningful results. And I think that there has to be some consideration for making sure that you're not limiting animals too much, so that you're getting results that can't be replicated. And I think scientists don't necessarily do a good job always about thinking about exactly how many animals they need for a study. Obviously, you can think about that in advance, and given the effect size you're expecting to see, you can decide how many animals you need to use. But it's a difficult issue because the ethical considerations about using animals are important, but so are concerns about using enough animals to getting results that are robust.
All right, there is a comment here. It would be also very interesting to hear your perspective about the role that scientific journalism in these issues. How are journalists also serving the information that get around to the more general public?
Yes, science journalists are certainly part of this problem, partly because we are part of the reason that people want to publish flashy findings is that then they will get attention in the press. And so that advances their career if they can have an exciting finding. The other thing that journalists focus on are exciting and unexpected results. I mean, we care about news, right? So things that are exciting and unexpected make news. The problem is we don't always or often follow up. If a single study comes up with a very exciting result, we may report that, but then if 10 other studies show that it's not true, we may not even notice it's 10 other studies. So we also create a distortion, and we need to be very careful about that because, in fact, that's the serious part of this problem as well.
Okay. Another question here from the public: has PubPeer contributed to the transparency in scientific work?
I'm sorry, I didn't hear the question.
Has PubPeer
PubPeer, yeah,
contributed to the transparency in scientific work?
Yes, I think it has. PubPeer is, for those who don't know, is a website. I actually haven't looked at it for a few years, and I assume it's still running. But PubPeer is a place where you can post anonymously questions and comments about research, and I think it can be a very valuable way, and it sometimes has exposed people who are manipulating their data and so on, and they've been exposed in PubPeer. But also because it's anonymous, I think it's also concerned to make sure that people are actually not just going after their rivals, but are actually raising legitimate questions. So I think it's not a very reliable source of information, but I think it can be valuable if read carefully to identify potential problems in scientific research, particularly things like image manipulation and things like that may be exposed on PubPeer.
Okay, somebody wants to ask directly a question to you. Nicolás.
El profesor Nicolás Pineda tiene la mano levantada, no sé si desea realizar una pregunta. Y también hay otra pregunta en el chat, profesor.
Sí, gracias. Yo quisiera preguntarle al profesor en inglés. Hi, Professor Richard. Thanks for your interesting talk. Of course, manipulating data is not fine. And regarding the research studies that would only express a hypothesis once the data has been analyzed, that brought to my mind the topic on free hypothesis studies. And I would like to ask you, what are your thoughts on that type of studies? I mean, on free hypothesis studies.
Yes, that's a complicated question. I think that there are instances where that may work in particularly genome analysis and so on. I'm not an expert in this topic, so I can't speak authoritatively about it. But I do think that that there are other paradigms for pursuing science, but I think they have to be done very carefully and not present them as if they were the standard that we're used to, where you develop hypotheses, gather data, collect it, and then analyze that data. But I think I think it's important to be open-minded about other ways of approaching science, but also to be skeptical about ideas that are or very cautious at any rate about things that are not done through the standard methods of science.
Okay.
Thank you, Professor. I have another question. Perhaps just missed that point. And it was on what happened in the end with the person who got the ALS diagnosis.
He was only able to participate in one clinical trial because with ALS, your health deteriorates, your health fails over time. And so the vast majority of people with ALS die after within 4 or 5 years. And I'm afraid that was the fate of Mr. Murphy.
Thank you very much. I don't have any more comments or questions.
Somebody wanted to ask another question. ¿Sí me están escuchando? Sí.
Sí, se oye en el chat.
Okay. Richard, what? Excuse me, are you listening?
Profe, hay 2 preguntas más en el chat. alcanzó a leer usted hasta la pregunta de Juan Esteban Pérez. Hay una
Sí.
pregunta de Wilson Daniel y una pregunta después de esa. Hay dos preguntas más en el chat.
Okay, Wilson Daniel dice, Richard, thanks for your talk. What do you think of biohacking to democratize science?
Could you repeat that question?
Yes, thanks for your talk. What do you think of biohacking to democratize science?
Biohacking to democratize science. Well, that's a big question, isn't it? I think that we really need to, as a community, think carefully about making sure that results are not only done safely, but that our scientists, studies are done safely, but that results are actually believable. And I think that biohacking, as I understand it, could be pretty free form. And I think we need to be very careful to make sure that people have got to the conclusions they think they have and doing their science safely. So I have some concerns about that. But I think that the underlying concept, I think the questioner is asking is if many people are doing experiments, that's another opportunity to validate findings if people are doing the same experiments. But then that gets back to the question of whether they're publishing their negative results as well as their positive results, and whether we can really accept those findings at face value. I think that's an issue.
Excuse me, and also you have to think of how innovative is your question or what you're going to do. And if it's not innovative, nobody's going to pay for it. So that something that the economics of this that you have already mentioned. I have a question. What percentage of the literature, of the current literature today, would have to be sent to the trash can if you were going to start anew?
Yeah, well, I'm sure everyone would love to be able to answer that question, and I don't know exactly how you would approach it. I mean, clearly there are some studies involving cell lines that I think probably a significant proportion of those need to be treated with a great deal of caution. I think that in the early days of genome research and analysis of genome research, a lot of that science was also very poorly conducted. But in recent years, that has improved. So I think if you look at more recent data, I think, and more recent findings from these massive genome examination studies, they've figured out how to do it more credibly. And I think a lot of those studies are probably pretty good. The same thing actually happened in clinical research involving human beings until the last 20 or 30 years. A lot of that research was also done very, very poorly. And there was a movement to actually make that better. And one key factor was they started asking people to publish in advance and state in advance what their studies were. And you can find those on clinicaltrials.gov. That way you can't manipulate and change your study midway through it, or if you do, you have to acknowledge that you've changed your study. There's more concern about looking at the statistical power of those studies as well. So I think there's still room for improvement for sure in clinical trials, but clinical trials have improved a lot over the years as people recognize the same sort of problems in the 1980s and 1990s. There's some talk about applying some of those same ideas to preclinical research and asking people to publish their hypothesis in advance before they do their study, so that to reassure people that they aren't changing the study midway through or or cherry picking their findings or whatever. So I think we've seen real improvement in clinical research, and it's obviously more cumbersome than just doing experiments in the lab, but there are people who advocate for doing that for preclinical research as well.
Do you think that it's going to be the need or the space for repeating at least some of the experiments on which you are basing your new work, your new hypothesis?
That's obviously an issue, is to make sure that there's funding for that, and there often isn't. If people do this, or at least try informally, if you're a graduate student and there's an interesting idea somewhere else in the literature, your professor might say, see if you can replicate that finding, and if you can, let's work on that study, on that idea. And so this does happen as part of on studies that aren't ridiculously expensive. Some of this replication does happen even though people don't apply for a grant in particular to do that particular piece of research, but it's part of the education process and part of the way an academic lab runs. So some of this does happen even though there is not targeted funding, but clearly I think for important and interesting findings, funding agencies should be more open to giving money to other researchers to try to replicate an important result.
Okay, the BRIM says, are there some kind of requirements to coach PIs, principal investigators, I suppose, to control their ego and greed? We are all human, prone to that, and we do not realize when we have these issues.
Yeah, I don't think there is a formal program to do that, but in some of the cases of outright fraud in science in the United States are often traced back to postdoctoral researchers in very highly competitive labs, because postdocs realize that if they don't get a really flashy paper during the time that they're working as a postdoc, it's unlikely that they will be able to pursue a career in science. And so they get into these highly competitive labs. Then they may manipulate or even make up data sometimes, and that is a major source of outright misconduct in science, in biomedicine in the United States. It's not a big part of reproducibility. Most people feel that most of these problems are not outright fraud, but a few percentage are, and that is a serious concern. Is these postdocs in these highly competitive labs who basically their career depends upon getting some exciting finding.
Okay, one more comment. The problem is also collaboration. Good collaboration is defined by papers, and the collaborators require papers. Now, Jorge Mario says, Richard, thank you for your talk. Regarding the Duke 40 scandal, maybe the main example of these bad practices in biomedical research. Can give us your thoughts on what measures have been taken to avoid this kind of a scenario?
Yes, I think that is certainly an example of outright misconduct, not simply people cutting corners or trying to work around the edges. Clearly in the United States, people are concerned about this, and there's institutions that are supposed to be looking after misconduct, but unfortunately, there are very few investigators. Very few people get caught like this every year, and universities may have incentives not to admit that their own professors are engaged in these problems, and the federal government, which is funding these programs, doesn't necessarily. It doesn't really have enough personnel to really investigate. Very few of these actually end up getting investigated. So I think the potential is always there for cheating in science, as there is anywhere else in human society. But we have to hope that scientists, by and large, are, and I believe they are, motivated to help to really explore what's happening in the world and to help people solve real problems. So I think that there will always be people who break the rules, and we need to make sure that, to the best we can, that they get if they're, seriously misbehaving, that they get caught. Does not always happen, but it should happen more.
Okay. Now a comment brings is Claudia Trujillo. Welcome, Claudia, and thank you for your participation. She says PIs put pressure on postdocs with visa in USA and put a lot of work on them. Moderate to severe anxiety is a big problem in graduate students. And yeah, that's all with her. But thank you for your very interesting talk and discussion. Thank you, Richard. I enjoyed very much your book. I felt I felt somehow supported. I have another question, which maybe we are reaching the end, which is bad. I was kind of happy with so much yet to talk about, but well, definitely I want to be your friend, Richard, but I want to ask you, in these 5 years after your book, have you won more friends than enemies?
I was surprised when I was reporting the book. I thought I would make many more enemies than I did, but I was surprised how many people really wanted to talk to me about these issues, because scientists see this as a problem in their world, and they see the value of bringing it to light. So I was surprised that I made more friends than enemies when I wrote the book, and the reaction from scientists once the book was published has been much more positive than negative. I think yes, there are some scientists particularly object to the title of the book, and because it seems to imply that science is dead. I actually talked with my publisher about that title a bit. It was not my suggested title. I wanted. I had other ideas for a title, but I think that once people get past the title and read the book, they realize it's not a blind attack on science. I tried to make it a thoughtful and balanced look at what was working and what's not working, and focus on some efforts to improve the situation. So I think in the end, I think I made many more friends than enemies.
Well. Okay, well, congratulations again. That's a very good result. I wonder, Paula, how is our time?
No veo más preguntas.
¿Cómo va el tiempo?
Vamos, ya pues estamos redondeando, profesor, en el espacio de preguntas. Sin embargo, no veo como más preguntas en el chat. No sé si usted hizo la pregunta del profesor Jorge Mario. Creo que sí. Veo una.
De Lina María.
Lina, sí, que está pidiendo como un espacio ya como editorial, que son quienes, bueno, también quisiera aprovechar con las personas que se encuentran conectadas en este momento. No sé si hay más preguntas, no veo como más preguntas en el chat, entonces quisiéramos aprovechar con las personas que están conectadas en el momento desde la corporación a quienes nos acompañaron hoy. Vamos a hacer la entrega, una vez estén impresos los libros. Vamos a hacer entrega de algunos ejemplares impresos del año ya entrante entre las personas que nos acompañaron en la mañana de hoy en este listado que están nuestros participantes. Entonces mientras ustedes van como haciendo el cierre, les voy a poner en el chat un formulario para que por favor lo diligencien. Y entre estas personas ahorita voy a pedirle a usted, profesor, que nos regale unos numeritos para hacer entrega de unos libros entre los que se conectaron. Quieren, vayan haciendo el cierre, pero para que no se me vayan a desconectar, en unos minuticos pongo el enlace en el chat.
Okay. Well, Richard, this has been very successful. Thank you very much. I want to remind you of other compromises that you have, maybe not explicitly, but I'm giving you the time to do it now explicitly. We are very interested in our postgraduate group in creating other lines of postgraduate work and very particularly in transdisciplinarity or areas which can work or are interdisciplinary by themselves, like scientific journalism. We have at the university, we have found that there are some other initiatives, so we are trying to put all them together and definitely offer the postgraduate, our basic biomedical postgraduate program as a laboratory for students from journalism, and also offering our graduates in biomedical sciences to go into journalism and try to improve our basis on that area and our professionals, because we definitely need more on that. I think that is clear, and apparently is also a little bit on fashion on these days of pandemic. So we want you here in Colombia as soon as we could, and also we ask you please to think on how you're going to help us with the education of these scientific journalists in our group.
Well, thank you very much. I look forward to traveling to Colombia, and I do hope that that will be possible soon. I'd be happy to keep talking to you about these issues. It's been a pleasure meeting with you and your colleagues about these topics, which are obviously very important to me. So thank you for your interest.
Okay, Richard. Well, thank you very much. There is no more questions here, are there? No. So then let us to applaud your effort, your book.
Thank you.
Yes.
Vamos a dar el paso, profesor. Acabo de poner en el enlace, antes de que el profesor Richard se nos desconecte, acabo de poner en el chat el enlace para que entre las personas que estén acá vamos a hacer entrega de 15 libros. Ya pues cuando la editorial los tenga impresos se los haremos llegar, entonces requerimos que todo el que esté interesado por favor diligencie, mientras la profesora Lina desde la editorial Biogénesis nos va a dar un saludo de cierre, para que por favor entonces todos se estén diligenciando. Una vez terminemos con el saludo de la profesora Lina haremos como el sorteo acá entre todos de estos libros.
Dr. Harry, it's a pleasure to meet you. I'm the director of the Publishing House Biogenesis, and on behalf of the Publishing House and the University of Antioquia, we want to thank you for your work and the possibility of having it translated into our language to continue with dissemination of these important issues. I don't know if you know, but our publisher has a free download policy to improve access to knowledge. I'm sorry for my talk. Sorry. After finish some legal details, we hope to have your book on our page and invited everybody to know it. And thank you so much.
Well, thank you so much, and thank you for your interest in my book. I appreciate it.
Thank you, profesora Lina. Muchas gracias, profesor Jorge, por acompañarnos en esta pequeña presentación, introducción al libro de Richard Harris. A todos también por acompañarnos. Voy a hacer el cierre. En este momento creo que ya todos los que estaban interesados diligenciaron el formulario. Vamos a hacer entonces el cierre del formulario e informaremos acá de una vez quiénes tendrían los libros. Como se los indiqué ahorita, no alcanzaríamos a entregarlos en el año 2021. Estaremos haciendo entrega ya en el 2022, pero por eso pues pedimos como sus datos de contacto. Permítame un segundo. Las personas diligenciaron y ya les informo quiénes tendrían el libro impreso ya con la traducción al castellano. Profesor Jorge Osa, ¿me escucha? 21 personas diligenciaron el formulario. Vamos a hacer entrega de los 21 libros a todas las personas que diligenciaron el formulario. Les estaremos enviando un correo. ¿Me escuchan?
Sí.
Ok.
Ah, todos ganaron.
Todos ganaron. Vamos a todo el que diligenció el formulario, le vamos a enviar un correo electrónico con el bono físico, virtual, perdón, para que ya estén atentos el año entrante para la entrega de los libros. Muchas gracias al profesor Richard Harris, muchas gracias al profesor Jorge Osa, a la profesora Tatiana, quien estuvo acompañándonos también tras bambalinas en esta transmisión, y a todos ustedes por estar con nosotros en la mañana de hoy, por acompañarnos en la conmemoración de los 25 años de la Corporación Académica de Ciencias Básicas Biomédicas. Les recordamos que tendremos también conexión en el evento central a las 3 de la tarde. Los esperamos a todos. Lastimosamente, por temas de aforo, no podremos contar con la presencia física de todos ustedes. Sin embargo, para nosotros es muy importante poder tener este acercamiento y contar con todos ustedes el día de hoy. Los esperamos entonces también a las 3 de la tarde. Nuevamente, profesor Richard Harris, thank you. Desde la corporación, muchas gracias por estar con nosotros, por abrir esta conmemoración, por abrir este evento de felicitación. No podríamos abrirlo de una mejor manera que con esta grata conferencia. Muchas gracias a todos ustedes. Que tengan un excelente día, profesor Jorge, profesora Tatiana. Muchas gracias también por estar con nosotros.
Hasta luego.
Yes.
Thanks again. Bye-bye.
Bye.
Bye. Okay.